Hello, everyone. I think it's time to start. Hi, everyone. Well, let's start with the process first. For all the audience, we're going to mute your lines. If you have a question, please use the Q&A box in the bottom of your screen, and we will be recording this webinar so we can share it later on. Welcome to the amazing webinar that is hosted by AFAR, American Federation of Aging Research today. The subject is super exciting. I was looking for that for many weeks while we were preparing that. It's about bioprinting, organ regeneration, and beyond. We have an amazing constellation of stars and talents for you today. Yeah, I'm moderator of this amazing conversation. Sergey Young, AFAR board member. I'm a longevity investor through my Bold Longevity Growth Fund and the book author. Our host today is American Federation of Aging Research. For more than 40 years, AFAR has been committed to advancing the science of healthy aging. AFAR has served as a field talent incubator, providing more than 200 million to a little bit below of like 4.5 thousand investigators at different research institutions to date, and the number is growing. And AFAR also work with public and private funders to support high quality grant programs and research networks. Let me start with a short intro of amazing scientists and entrepreneurs who are joining us today. I'll start with Eduardo Chini. It's close to my heart as a AFAR board member. Eduardo is a AFAR grantee. He's Associate Director of Kogut Center on Aging. And what is also important, he is part of Transforming Transplant Initiative by Mayo Clinic. Welcome, Eduardo.
Thank you.
The next one is Michael Hufford, co-founder and CEO of amazing company, one of the rare in the space called LyGenesis. He's based in Pittsburgh. Warm welcome to Michael.
Thanks, Sergey.
Thank you. Michael Levin, scientific founder of Morphoceuticals and director of Allen Discovery Center and Center for Regenerative and Developmental Biology at Tufts University.
Thank you so much.
And Tom Rando, AFAR President and Director of Broad Center of Regenerative Medicine and Stem Cell Biology of UCLA. That's it for the intro. Let's go to conversation. And the first round of the questions that we have for you today, I'd like to start with Tom. Tom, we went a long way in terms of advancements in bioprinting and organ regeneration in the last few years. So what do you think are the most significant emerging breakthroughs in the field, and why is that important for the new therapies and technologies that we will discuss today?
Thank you, Sergey. It's a real honor to be here, and this is a topic very near and dear to my heart. I've worked on sort of stem cell biology, tissue regeneration, organ regeneration for much of my career. And asking about the advances, we could spend the hour talking about the advances in recent years in this. And I think we can easily point to some that have really developed in the past couple of years. Maybe to get going, it might be worth just having a couple of concepts and definitions, because I think we'll be hearing different kinds of words and phrases. So first of all, what I think of so miraculous about this field is really what we're trying to do is recapitulate what every embryo of every individual does from the beginning. Take a group of cells that may have no features at all and turn them into livers and pancreases and brains, and that's just a miracle of modern biology. And we're trying to capture that and kind of recapitulate that in many ways, sometimes even make it better, which is really challenging. Part of that field has really been based on this notion of so-called induced pluripotent stem cells, or iPSCs. And those are cells that we can now create, and this is work that received the Nobel Prize. We can take cells from an individual, say a skin biopsy, and turn those cells back into kind of embryonic cells to have no features. So then those embryonic-like cells, or the iPSCs, can then be induced to make all these organs and all these tissues, blood and heart and brain. So we have the ability now to kind of recapitulate that developmental program through these iPSCs. And that's been an amazing technology that has, really been rampant in the field. And the other more recent that I think has also been transformative is the technology of so-called organoids. So what are organoids? So organoids, it has been found empirically that if you take some of these stem cells and put them in a dish, they will start to make these organs. They'll make different cell types, and they'll make little guts, they'll make little brains. And so already within the kind of biology of those stem cells, there's the capacity already to start down this path of making new organs. So I think we'll hear about these different kinds of technologies. What are the advances in recent years? I think one that comes to mind is, for example, using iPSCs, these pluripotent cells, to make differentiated cells for organs that don't regenerate, like the heart. So it's now possible, we're getting closer and closer to this with some trials, where these iPSCs in the laboratory are turned into heart cells and then transplanted into a damaged heart, like a heart attack. You have a heart attack, the heart doesn't repair, but it's looking more encouraging that we can take these in vitro, these in culture differentiated cells, transplant them, and then start to form, heart around those new cells, so that they basically engraft and incorporate into the heart. So that kind of taking cells and then transplanting them is one of the real advances, I think. And finally, and then I'll turn over to my colleagues, but another one, and I think this probably relates to a lot of the biology that others study, is can we in fact build organs in a dish? So I mentioned these organoids that already start to form little hearts, little brains. Is it possible, and this is where bioprinting really comes into play, can we print the structure of a kidney and then put the stem cells on that kidney, let them form that kidney, we direct it, and then have an actual kidney that we transplant from the dish into the person? So, the first one I mentioned is more of a cell transplantation, and the second is actually building organs in the culture and then transplanting them as whole organs. And that's really critical because a lot of things that are missing from these say organoids is the blood supply, and every organ has to have a blood supply. But bioprinting has become increasingly adept at, for example, developing the vasculature within an organ. So I think we'll hear this from the other panelists as well, is how bioprinting and regenerative biology have really come together to advance this field of organ regeneration.
Amazing. Thanks, Tom. The next question goes to Michael Levin. And I'll start with a little bit of the context, Michael. So a few years ago, we all woke up in a completely different world, in the world of AI. So, and finally, whenever I think about this, I think that finally, through the whole history of evolution of humanity, we have something which can help us to handle the complexity of human biology, which is artificial intelligence. So for the field, how do you envision integration of AI in advancing organ and tissue regeneration?
Well, in order to talk about my favorite application, I want to take a quick step back and just look at the kind of like what Tom just did, a look back at what we're actually trying to solve here. So if you think about a salamander, so salamanders regenerate their eyes, their limbs, their jaws, their tails, including spinal cord. And what happens is that if they lose a portion of the limb anywhere along the proximo-distal axis, the cells spring into action, they rebuild, and then eventually they stop. When do they stop? They stop when a correct let's say axolotl limb has been completed. And so this is very important because it's a kind of homeostatic process whereby the system can tell that it's been injured, it knows what to do to rebuild, and then it knows when to stop. That last part is really critical, because one of the things that leads us to ask about is, how does it know what to do? Okay, what is the stop condition? How does it know what direction to travel in this anatomical space of possibilities? It could build many things. And so one of the things that we and others in this field have been studying is the role of bioelectricity. So if we have a system that seems to know what to do, to take steps to do it, and then to recognize that it's been done and stop, how could a group of cells do that? And we have a very good example of this in the brain. So in the brain, it's everybody knows lots of different animals can store memories. They can pursue those memories as goals, and then they can cease. When they've achieved it, they stop. So years ago, we started to look at this question of what could possibly encode these pattern memories. And one of the things that our lab and others in this field of bioelectricity have done is to develop techniques to read and write those pattern memories. So there are now visible, we now have techniques, much like neuroscientists try to do this in the brain, to read out the electrophysiology and translate it into, well, what does the animal remember, what is the patient thinking about, and so on. We now have the ability to read those patterns, working on decoding some of them, and we have the ability to rewrite them. And so this is really interesting, because when you do this, when you rewrite these patterns, you can do things like induce whole ectopic organs in other locations of the body in animal model systems. This is not in human use yet, so we can make ectopic eyes, brains that trigger limb regeneration, fixed birth defects. These are all things that have been done. And so here's the interesting problem that we face. If we have a situation that is not what we want it to be, meaning it is either not regenerating or it's making the wrong pattern or something like that. The question is, what kind of bioelectric pattern do we have to incept into the tissue, okay, in order to get it to do what we want it to do? In other words, the bioelectric layer is an interface, just like we are now communicating through the bioelectric interface to our neuroanatomy. And so the question is, what do you want to say through that interface to get the tissue to do what you want it to do? So here, to me, is an incredible opportunity for the AIs that are being developed now, which is to help us crack this code and solve this. This is fundamentally an inverse problem. We know when you have a particular pattern, this is what happens. What pattern do you want to have to make a limb, an eye, or whatever else? And so using AI to reverse that process and to say, if I want this pattern to be a certain way to regenerate a limb, let's say, what is the drug that I want to turn on and off specific ion channels? Which ion channels are those going to be to establish the correct state and get it to happen? And just to point out that the fact that I think at last time I looked, something like 20% of all drugs are ion channel drugs. And so that means that we have an incredible domain of electroceuticals. These are ion channel drugs, usually used for other things in the nervous system, in the inner ear, gut, all these things. And so all of these things are potentially available if we had an AI system that would say, this is the channel, these are the channels you want to turn on and off in this situation. And thus, these are the drugs that you want to use. So for me, the ability to crack that code, and of course, it's not just bioelectricity. It's also, it will be useful. The same thing will be useful for biochemical signals, biomechanical signals. But that's my favorite application.
Amazing. Thanks, Michael. The next question goes to Eduardo. Eduardo, you are an essential part of Mayo Clinic's Transforming Transplant initiative. Can you tell us more about the initiative itself, and what are the most notable developments in the field coming out from that?
Yeah, so as mentioned on the beginning, so I also, in addition to run a basic research lab in the basic biology of NAD metabolism and aging, I'm also a clinician, and I do, as an anesthesiologist, mostly transplant, especially solid organ transplants. And I was in Rochester, Minnesota, came to Mayo Clinic, Florida, essentially because about 4 years ago, Mayo Clinic, Florida started this program called Transform Transplant. Okay, and number one, I think it's important to highlight that right now, Mayo Clinic is the number one provider of transplants in the US. So we have the highest volume of anybody else, with amazing outcomes. But there's still a lot of challenges, right? The main goal of the Transform Transplant in the future is that no transplant will ever have to happen again. So that is the goal of Transform Transplant. You'll be amazed how surgeons will be happy not to transplant an organ if we can avoid that. There are many, many steps to get there. So there's actually a collaboration between Mayo and CMU to develop new devices, to develop sensors and things like that, right? If you think about aging, and that's where I actually came from, because as the associate director of the Aging Center, when I came here, I identified that the Transform Transplant is where we should collaborate. And I'll tell you why. So as Tom said, there's all the development in regenerative medicine, organoids, and there is an understanding, I think, of investigators and the NIH that you should start to go into organoids. And our idea was, what better than organoids than actually an organ, right? So what if we can start to manipulate the so-called hallmarks of aging in actual human organs? Okay, and how do you do that? I think it's that part we are already almost there, and I think we figure out things over the years. So also to give you this background, the number one cause for a dysfunction of an organ after the transplant is the cold ischemia, how long it's ischemic. The number two is the age of the organ, right? So if the organ comes from an older donor, it's less likely to work well, okay? So how do you fix that problem? One of the, in my view, one of the biggest changes that I think will change the way we can approach the biology of aging and understand more about those interventions, and also to have a huge impact in transplant, is the fact that now we can take an organ, okay, a liver is a great example, and perfuse that organ ex vivo for about 2 weeks, okay? Imagine what you can do on those 2 weeks. The window is enormous. So the idea of the collaboration between the Aging Center and the Transform Transplant is that we will take organs to a facility for ex vivo perfusion, and we will rejuvenate organs ex vivo, okay, through intervening at the hallmarks of aging. So that goes all the way from metabolic reprogramming to partial reprogramming, obviously senolytic approaches and things like that. And in fact, there is data demonstrating that if you actually do senolytic therapies in a liver ex vivo, you can actually prevent some of the dysfunction that happens on the biliary tract when it's perfused. So the way that we sold, especially to the people that are putting investment on this, is that this will come a time where regenerative medicine will be your own organ. So imagine a time that you have a kidney that's not working well, we'll take it out, you're on dialysis for a couple of weeks, we fix your kidney, and then we give it back to you. And now you have a young kidney, a young liver, a young organ you may. If you put then CAR T cells, for example, to clear senescent cells, you might be able to not only treat their organ, but then those CAR T cells are back to you, clearing senescent cells in other organs. So I think that that's sort of the bold vision. We do see that the organs are going to be the ultimate organoid, where we are going to learn a lot of those interventions and actually hopefully transform the way that transplant is done by augmenting and rejuvenating organs ex vivo for transplants.
Thanks, Eduardo. Amazing. And the next question goes to Michael Hufford. Michael, LyGenesis is a pioneer in using lymph nodes for organ regeneration. And I know you are already in a clinical trial stage for liver diseases. I guess two questions. One is, how can you use it to solve the ongoing organ shortage crisis? So that's one. And second, what are the other organs that you have in your development pipeline, and why?
Great. Well, first, let me thank AFAR for hosting this. It's really wonderful of you all to put this on, and great to be part of the panel. Great to see you again, Sergey. So let me start by way of kind of background for folks that may not be familiar with LyGenesis. So LyGenesis is an organ regeneration company, and we're leveraging the body's natural bioreactors, which are lymph nodes. So throughout your body, you have 500 or so lymph nodes spread throughout your body that evolution have developed as an incredibly efficient bioreactor for T cells to help us fight infection. But as many people know, if you develop cancer and cancer finds the lymph node, there's a sense that's a bad thing. And it's a bad thing because a lymph node will bioreact all sorts of tissues, including cancerous tissues, to form tumors. And the fundamental scientific discovery behind LyGenesis was my colleague Eric Lagasse here at the University of Pittsburgh, who, thanks to a decade of NIH funding and hard work he had put in, he discovered that you could bioreact a variety of different tissues within the lymph node. And in fact, in particular, and we'll start with the liver, you could form a fully functioning ectopic liver by transplanting the key liver cell, the hepatocyte, into a lymph node, where it will remarkably overtake that lymph node. Eventually, the lymph node disappears, and what you're left with is histologically identical to the native organ. You're left with an organ that, one, is well vascularized, that contains liver lobules, and we found in a number of preclinical studies would rescue animals from otherwise fatal liver disease. So that was sort of the scientific discovery. What we have going for us as now a clinical stage cell therapy company, and I should note, Sergey, we would not be here without the support of Bold Longevity. You were a very early investor in our work, so deeply appreciative of that, along with Juvenescence and now Prime Movers Lab. So we're now a clinical stage company. Among the things that are exciting about this approach is that clinically, you have 50% of patients don't even qualify for an organ transplant because in the case of end-stage liver disease, they're simply too ill to withstand the procedure. So critically, we're using an endoscopic ultrasound procedure where we go down through the mouth of the patient. And in a 10-minute procedure, we're engrafting our cell therapy into lymph nodes adjacent to their liver. So this is a 10-minute procedure done under light sedation. So that means that a number of patients, a huge number of patients, who would not otherwise even qualify for an organ transplant and would unfortunately suffer and die from their end-stage liver disease, now have the potential for this therapy that's now in development because of the low-risk way that we engraft the cells. As a biotechnology company, you're always worried about regulatory, and so from an FDA perspective, because there's a rich history of full organ transplant, there's a lot of good precedent for what we're doing. And lastly, to answer your specific question, Sergey, from a commercial perspective, so each donated organ, a donated liver around the US, about 20% are unmatched currently, right? They can't find a match for the organ. Well, each liver is made up of billions of these hepatocytes. But in our clinical trial, we're engrafting only a few 100 million of those hepatocytes. And what that means from a supply and demand perspective is that today, one donated organ treats one patient. Using our approach, one donated organ could treat 50 or more patients. So really upending the supply-demand calculus when it comes to this 1-to-1 ratio today becomes 1 to 50, right? And when you combine that with the cost of a full liver transplant, about $1 million today, the cost of an endoscopic ultrasound, about $2,000 today. And when you think of being able to use a single liver to treat dozens and dozens and dozens of patients, that's when it becomes very exciting from a clinical, commercial, and just human perspective. Lastly, I'll just say Eric Lagasse's research that he published in Nature Biotechnology years ago showed the lymph node is very agnostic as to the tissue type. So it will grow fully functioning ectopic livers, which is our lead clinical indication. But he showed that you can put kidney fragments in a lymph node and it will generate an ectopic kidney that will generate urine. Critically, from an aging perspective, an ectopic thymus is incredibly exciting, where you can take an older animal and graft a young thymus into that lymph node, and that will form a functioning thymus and start educating T cells, has important implications for aging in our immune system. And lastly, pancreatic islet cells responsible for diabetes also thrive in the lymph node where they glucose sense and insulin secrete. So it's also a potential pipeline therapy down the road for diabetes.
Perfect. Thanks, Michael. Just a reminder to our audience, please feel free to share questions. So far, we have one question asked. I couldn't believe it. So what we're gonna do a second round. There was like optional questions that we prepared in AFAR for our panelists. This time, let's try to make it a little bit shorter. Thought I can, listen to each of you forever. So we have another like 15 minutes in the end of the hour for Q&A from the audience. Okay. So I'd like to start with Michael Levin. Levin, as it is about how electroceuticals can be used. And then, so the question is regenerating lost limbs, regenerating organs in humans, some age-related conditions like neurodegenerative diseases or muscle atrophy. So what's your vision for that?
The thing that we are focused on, and so in particular at the company that David Kaplan and I are scientific co-founders of, which is Morphoceuticals, we are specifically focused first on limb regeneration. So the idea is to trigger in mammals and eventually human patients the kind of limb regeneration that you see in other organisms. So it's a combination of wearable bioreactor that sets the environment, and then the payload, which is these electroceutical drugs to trigger the process, not to micromanage it, but to serve as an initiator trigger at the beginning.
Thank you. The next question to Tom Rando. Tom, you've done extensive research in muscle stem cells and regeneration, and I think it's particularly valid topic in the context of arrival of GLP-1 drugs, because one of the, it's many benefits, but one of the downsides is people lose muscle mass. So how close are we to regenerative therapies that could actually help older adults regain some of their lost muscle strength or organ function?
So that's a good question, Sergey. I mean, we study muscle stem cell regeneration mostly in the context of either degenerative disease where muscle is actually lost, or trauma where muscle is lost, as in blast injuries to soldiers. There you have basically a context where stem cells can be implanted and then can start to form new tissues, much along the lines that other panelists have talked about. So that's a regenerative therapy, and we've been successful in that in small animals. The big challenge in humans is scale. We go from a small animal to a large human. It's much, much bigger, and so we have scale issues for the matrices that we use, for the number of cells we need. So that's for regenerative therapy for injury, say, like one has in a heart during a heart attack. One gets a trauma to muscle, you lose muscle. What you're referring to is more of muscle wasting, and that happens with the GLP-1 drugs. It happens in cancer. It happens with age. We call it sarcopenia. The biology of muscle loss in that context is somewhat different. And what has not been as encouraging is the use of stem cells to maintain or restore muscle mass in the absence of some kind of trauma or disease. And so I think that's really kind of at the edge of our understanding of the relationship between the kind of regenerative biology we're talking about and the kind of atrophic or wasting condition that leads to these. I am hopeful that there will be a connection, that there will be the possibility of using regenerative therapies. But I think at this point, we're a ways away from thinking of using stem cells, for example, maintaining muscle mass in a GLP-1 agonist person taking that, where I think other drugs, actually drugs that preserve muscle mass, are more likely to be effective.
Very interesting. Thank you. Let's make it a little bit more interactive. We have more activity in our Q&A. So I'll come back with a few more questions to Eduardo and Michael Hufford. Saul Rosenberg, thanks for your question. It's very challenging for me, a little bit technical, so we're going to come back to you later on after the webinar. Mark Velichkov, I apologize if I didn't pronounce correctly. The question is, who is researching the psychological and ethical implications of these breakthroughs? I think it's a very important question. And I actually, my own fear, through our investment work in both longevity growth, is we can be ready for a lot of breakthroughs technically or scientifically in 10 to 20 years from now, but we might face the barriers in terms of the ethics, acceptance from society, and acceptance for regulators. So I would welcome your views on the ethical side of it and psychological side of it. I don't know who's gonna start first.
Take a stab. I mean, for sure, I think the society will welcome regenerative therapies for repairing injuries, treatments for a heart attack, treatments for a liver disease. I think it becomes much more challenging in other parts of this field. For example, forming brain, forming human brain. There will be questions about what the limits are between the biology and then the actual issue of transplanting cells into a brain, maybe changing a person at the very fundamental level of their nervous system, who they are. So that becomes I think, a very clear ethical place we have to be careful as we go. One of the fields that is at that border of kind of biology and ethics is generating human embryos. That's clearly, there's a potential there for incredible amount of biological and therapeutic applications, but that becomes a very ethical issue as to how long we should be able to, in a culture dish, generate a human embryo-like structure. And so those fields are advancing along with ethical considerations at this time even.
Perfect, thank you. The next question probably goes to Michael Hufford from Brendan. How long does it take to grow a fully functional liver? And I think we have a sub-question from Gania Shabbat asking, how is that different, rather than working with these sick liver fixes for liver disease, how does it different to work on a new healthy liver?
Sure. So I'll say all of our preclinical data suggests it takes about 2 to 3 months to grow enough functional mass to support the diseased liver. And so our target of our clinical work, which is ongoing at Houston Methodist under the guidance of Drs. Mark Gobriel and Constance Mobley, transplant surgeons there, we're focused on patients with end-stage liver disease currently. And so that's the current application, not like acute liver failure. So, this is not a therapy that exerts its effects in days or weeks. It's a therapy that potentially exerts profound effects, but over the course of months. So that's what I would say in terms of the time course. And there is a video on our website that concisely summarizes the biology. In general, I would say the liver is unique insofar it is the only organ that naturally will regenerate. And so we've known that, for even back in the Greeks with the myth of Prometheus, and the liver would eat his, or the eagle would eat his liver each night and it would regenerate. So we've known for thousands of years that the liver regenerates. So a diseased liver is emanating many pro-growth signals. And the hepatocytes that we're engrafting into the lymph node recognizes those signals, and it triggers it to form these ectopic organs that basically offload some of the blood filtering that the diseased organ is now unable to do onto these ectopic livers.
Perfect. Amazing. The next is, what are the current challenges in bioprinting organs like the kidney or any other organs bioprinting wise? What I've heard, and it might be not the case in today's world, but just a few years ago, what I've discovered that majority, more than 90% of bioprinted organs actually, make it to the lab rather than to humans. I mean, is it the case today?
Yeah, so I think it is still very challenging, and you can even think about the most simple organ. So a bladder, right? You would imagine you could have bioprinted a functional bladder at this point and have it in use already. And even that is challenging. I mean, the biology is very, very complex. And when you actually take a kidney, for example, so the kidney has the nephrons that have multiple cell types organized in a very, very complex way, where each nephron actually connects its tubules with its glomeruli. There is the risk, obviously, of generating an isolated glomeruli that now doesn't have a tubule, and that actually causes problems by itself. So I think bioprinting is eventually going to happen, but I think we are still very, very far. I mean, as I said, we can't bioprint a functional bladder yet, and I think we are almost there, but it's a tough thing, and it is a question of time. We have through the history of science have solved many problems. I mean, if you think about the first transplant ever made, everybody thought that was crazy, and obviously the first patients didn't do well. You have the example of the xenotransplants recently that were a big breakthrough, but again, we're not quite there yet. And I think that this is also true for bioprinting.
Thanks, Eduardo. Just a couple of years ago, I had an amazing trip to see facilities where they work on lungs, set up by Martin Rothblatt and her team. So the question is, which is very relevant, I think, for that, it comes from William Wallace. Are there any efforts targeting the lungs?
I don't mind to take this one because actually here at Mayo, just across the street, they have what we call the lung farm. So there they actually take lungs before transplant, and they actually can monitor the function and sort of wait. You know, sometimes the lung is not that great, and then they are waiting for the lung to get better, and then transplant to a patient. The next level, next step is already coming, where you actually even get a lung, and there's a lot of work done in Toronto. You get a lung that is actually infected, right? So you wouldn't actually transplant that lung, but you put on this ex vivo lung perfusion system, you actually treat the infection, clear the infection, and then use that lung for transplant. So I think the power of ex vivo machine perfusion for organ optimization, organ regeneration, organ rejuvenation, and to really change the way transplant is done today can't be overlooked. I think it is the most transformative thing we've seen in the area of transplant. And even from the clinical point of view, it's much easier to take care of patients that had a liver transplant that was on a machine versus one that actually came on a box. So it has transformed. And as I said, I think it gives us the opportunity to try things potentially that we wouldn't try in a human being in terms of manipulating the biology of aging.
Perfect. Thank you.
Can I chime in there?
Please.
Yeah, I mean, again, the lung is well, every organ is fascinating, but the lung has, really distinct anatomical regions. And I think the beauty of this is where the biology comes together with the immunology comes together with the bioengineering. And so, for example, what was mentioned about the bladder being, a relatively simple organ and yet still very difficult to bioprint. Parts of the lung are simple, like the trachea, for example, is, one where bioprinting has made actually advances in developing a very stiff part of the lung. Doesn't apply to the distal part of the lung, the alveoli, where the gases are exchanged. But on the other hand, the organoids from the lung are exactly those kinds of alveolar sacs. So I can see the kind of these different technologies coming together in ways that will end up generating new organs by intersections of very disparate fields.
Perfect. Thanks, Tom. The next question, if I can address to Michael Levin, but please feel free to add as well. And it's an ethical and equality question, Michael. So are there any advancements in organ regeneration procedures going to be available to the working class people? So the question here, I've heard the figure of $1 million mentioned in liver regeneration. And I was thinking, how accessible would this actually be?
Yeah.
Thanks, Michael.
Yeah, thanks. No, definitely very important question. So I can address it from the perspective of the bioelectric side of things. Most of the reagents we use are existing drugs, many of them off patent. They're already used for other things. These are very cheap drugs. Standard electroceuticals are incredibly cheap and available. We use various antimicrobials, different drugs that are widely accessible to populations. And so I really have hope that at least on that side of things, and I can't speak to some of the other technologies that the others are expert in. On our end, these are not going to be expensive therapeutics at all. Now, that poses a bit of a barrier sometimes for the development side, because if these are drugs that are already widely available, right, the funding and so on, which is why we're so grateful for our partners at Morphoceuticals and others who are helping us push this stuff forward, because many of these things are indeed not expensive novel drugs, but already widely available therapeutics.
And I would just chime in too and add that, the current cost of a liver transplant is $1 million, right? So it is an extraordinarily expensive medical procedure. And you may have seen in the New York Times, there was a great story just the other week about the line for an organ and all the problems of managing organ availability. And I would just note, so I think the potential of iPSC-derived cells, bioprinting, and technologies like LyGenesis are to radically bring down the cost of and increased access. But I think it's a great question, because if you think like rain falls from the sky, but not everyone has clean drinking water. And so the extent to which you have remarkable new medicines coming out, I think we have to be very mindful about who has access to them, what do they charge. Those are, I think, incredibly important questions.
Sergey, I would add that, again, these are complex issues that you're raising in terms of accessibility. And there's sort of, who can pay for this just out of pocket because they have the money, who's insured but might not be covered, and who's uninsured. So those are really very different issues. From an insurance company point of view, which is always, important, a lot of these very expensive treatments, the benefit of them, even from the insurance company point of view, is they can be curative. And many of these conditions are very expensive to treat over the course of a lifetime. So it sounds like a lot of money up front, but in fact, and especially as costs come down, these can be cost savings to the healthcare system, because they can be curative as opposed to decades of chronic treatment, which can be extraordinarily expensive.
And if you don't mind, I would just add to what Dr. Hufford said. Let's say even if the technology is as expensive as a liver transplant, I think one of the things that we all need to realize is that the transplant is not a walk in the park. So a patient that goes through transplant will have years of immune suppression. And this is one of the things that I think also from that point of view, it's something that we really need to change, because patients that have a transplant have nearly 100% chance of having skin cancer over the course of their lives after transplant. So changing that by itself should be amazing. And also, the fact, if you can, by an endoscopic procedure, implant a liver or a kidney or whatever it is, you just save the patient a post-op course of pain, ICU stay, potential complications. So I think that all those technologies are just an amazing, and again, this is sort of our idea that we have on the transformed transplant, is really that what we do today should be viewed 50 years to 100 years from now as barbaric, essentially the way that we do it today. Right, if you think about it, and if you have ever been in the OR to see a transplant, it's not a pretty sight. So,
I agree. Thanks, Eduardo. Yeah, on affordability, we always got the affordability and accessibility question like through our work, because we support probably more risky, innovative, very expensive developments in the science and technology. And I'm really proud that almost every company that we support and other investors support bring in 5 to 10 to 20, not percent, but times improvement in terms of either quality of the intervention or the cost side of it. So I'm always been criticized for being like super optimistic, but I'm really super optimistic on what technology, a combination of human intelligence and artificial intelligence, can bring in terms of affordability and the quality of the outcomes for the healthcare. So we have a number of questions. I was trying to push back Saul's question, but it comes back from Thomas now. And there's a very good question from Judith, which I would like to like combine and reframe and bring a little bit of the brain angle, right? Neurodegenerative diseases like Parkinson's, like a brain work, what can be done externally from the outside in non-invasive way, what can be done internally from the inside in kind of more invasive way. I think it's a fascinating topic, and it's probably, you know, one of the most difficult organs, if not number one, to like regenerate or bioprint or take regenerative medicine view. So if I can open up discussion on that one.
Again, I didn't mention, but I'm actually a neurologist by training. These are very great interest to me. It's a challenging question. I mean, when we think of neurodegenerative diseases, they range quite broadly. Let's say from a disease like Alzheimer's disease, affecting large parts of the brain, to a disease like Parkinson's disease. And actually, Parkinson's disease is one of the success stories in terms of this has really gone from, the reason is because in Parkinson's disease, there's a very small area of the brain that degenerates. And so if one could regenerate that area, one could theoretically cure that disease. And that has been, you know, one of the leaders in this field of regenerative biology, initially using fetal cells from fetuses that can make this compound dopamine that's missing in Parkinson's disease, to now using these iPS cells to make dopamine and transplanting them into the very small area of the brain. And that is ongoing in human studies at this point. And so I think that that's going to be, you know, perhaps the low-hanging fruit in neurodegeneration that will see a success story in the context of regenerative biology. When you get to Alzheimer's disease, it becomes much more challenging and everything in between. But I think we at least have a handle on applying this in the context of diseases like Parkinson's disease.
I wanted to mention something that is not often kind of known, which is this question that I think Tom brought up before about significant regenerative events in the brain altering the patient and the memories and all of this. There's really good evidence from both invertebrate and vertebrate models that actually significant regeneration of the brain is compatible with the persistence of memories. In fact, in invertebrate models, the entire brain can be removed, and if in regenerative animals, when it regenerates, so do the memories. Okay, and so this to me represents a huge and very poorly understood frontier as to how information, both morphological and behavioral, moves through tissue, what other tissues are involved in addition to brains. And there is, at least I think, some reason to be optimistic. Of course, we don't know how it plays out in mammals, but it should be studied. And I think we have reason to be optimistic that significant regenerative events in the brain, once they're achieved, will actually be perfectly compatible with the persistence of the personality.
Perfect. Thank you. We have so many interesting questions. Unfortunately, I promised to our AFAR team to finish on time. Literally two, if we can just quickly do like two other questions. So one is what I learned, again, this is not my area of expertise. Actually, kidneys are like probably the most important demand-wise, you know, organ in today's world. So what are the efforts is currently underway? What are currently underway to solve the kind of kidney shortage problem?
I'll just say briefly on our side, so Eric has several papers that he's published showing that we can generate an ectopic kidney in a lymph node. That is a more basic research program. The kidney in many ways is far more complex than the liver from a regenerative perspective. We also say there's the non-trivial plumbing problem. So when you generate an ectopic kidney, you have to do something with the urea. So all those are kind of non-trivial problems yet to be advanced. I know there's a Kidney X Prize that's ongoing that's trying to fund a lot of really innovative work in kidney regeneration, but Eduardo, Tom, and Mike may be more familiar with some of those efforts.
Thank you. And the last one that we're going to take from the audience today, and it has a geographical perspective. Are there any other countries or geographies that are farther along in their research?
I'm not going to be an expert on this. I mean, I can say, I would not say that I can think of another country that is further along in research than the US in the context of what we're talking about today. There are countries where, rightly or wrongly, there are a lot more therapies being given as stem cell regenerative therapies, but I think they're unproven. And so I think, you know, while one can go and get stem cell injections, the proof that those are effective has yet to be shown in clinical trials. In terms of the technologies that we're talking about today, all the things that my colleagues have expressed, all these areas of bioprinting, regenerative medicine, there are collaborations around the world, but I don't think any country is further along than the United States in totality.
Perfect. Thanks, Tom. Well, that's a great finish with the compliments to AFAR and to the scientific and technological work of all of us. We have another 7 minutes, so can we just do like 1 minute closing remark, and I'll just do the like final 3 slides for the webinar. And for closing remarks, if you can highlight like most promising avenues for researchers, grantees, young scientists that they can focus on, or just give an advice to someone who would like enter in the field from scientific or entrepreneurial perspective. Well, let's start with Eduardo.
Well, so I think that the most important thing for anybody that gets into science is to be resilient. And I think we are experiencing this right now. You have to be persistent, you have to be resilient, and you have to follow the data. I think that those are probably the most important things to me. Don't be married to your hypothesis, be married to your data. And don't be afraid to be wrong. One of my first mentors used to say that if you are always wrong, it's because you're not doing something novel. You have to be wrong most of the time because you are trying to find something that has not been done before. So that is, I guess, my advice.
Thank you. Thanks, Eduardo. Amazing. Michael Hufford, please.
That's hard to follow. I would say focus on being at the intersection of fields, and I mean that both from a scientific perspective, but also just from a how to finance your research moving forward perspective. So I think it's so often the case that it is the intersection of fields where you get novel advances. I think Mike Levin's work in particular that I'm enormous fan of is a great example of this intersection of many different fields. So that's just from a high level perspective. I would also just notice someone that's on the industry side of things that, you know, there is industry can catalyze research and accelerate it in a way that is really quite dramatic over more traditional funding routes that may be increasingly needed, depending on how things play out moving forward here in the US. Regardless, I think I would just encourage you to kind of think about the ways in which industry financing can both be a bridge to the clinical application of your research, but also can open up novel areas of funding that can accelerate your research as well.
Thank you. Thanks, Michael. Michael Levin, please.
I would say I would echo what Michael Hufford just said about interdisciplinarity and not worrying too much about specific borders between tools and between disciplines. But just to kind of say one very specific thing, I think that the full solution to this problem and of transformative regen and complete regenerative capacity, I think is going to evolve the tools of cognitive science, computer science, and physics. And so I think my advice is look beyond biochemistry and genetics to the tools that are used to study the mind and to study computation and information, because a lot of what we need is actually traditionally from those fields, not just chemistry.
Thank you. Thank you, Michael. Tom, please.
So thank you, Sergey. And I think I would also echo what my colleagues have said, that I think, you know, when I think of the future of this kind of regenerative biology, I think of it at least as an intersection of transplantation immunology, bioengineering, and cell biology, developmental biology coming together. And that will be, I think, the wave of the future. You know, if there's one area that I kind of encourage people who are new in this area on the biology side, we haven't talked too much about, is really the area of synthetic biology, where we're not only giving back cells, but we're actually engineering cells to help them engraft and avoid immune responses and integrate all these ways that have proven to be, I mean, CAR T therapy that has been mentioned is just one of those remarkable successes. And, I just think, I run the Stem Cell Center here at UCLA, and one of my mantras is, I think at the end of this century, the different kinds of cell transplantations will be as common as organ transplantation was at the end of the 20th century. Something that was unheard of at the beginning of that century is a mainstay of medical practice. And we will be treating currently untreatable diseases with cell therapies and organ regenerative therapies in ways that were not imagined at the beginning of this century.
Thanks, Tom. What a vision. I'd like to live in this world with this vision. So before I'll cover 3 last things in the next 1 or 2 minutes. I just wanted to say a big thank you to everyone who joined us today, to the audience. Big thank you to all the participants of the panel. What an amazing conversation. You can watch this again or follow us for other webinars done by AFAR on the website, on the social media as well. Let's stay connected on the social media. AFAR is very active part of it. And finally, if you would like to support the work of so many amazing scientists and entrepreneurs through your donation to AFAR, feel free. It doesn't matter whether it's $100 or the $1,000 or $100,000. So let's help AFAR with that. And you can channel your donation through the AFAR website. Otherwise, thank you for your time today. Stay healthy and happy. Thanks, everyone.
Thank you so much.
Thank you.
Bye-bye.
Thanks.
Bye.